Thanks, Ryan. Yeah, so I figured I would just run through a broad overview of the study. We're sort of dabbling in a whole bunch of different things, and if there's particular things that are of interest and I should delve deeper into, we can do that. I think I promised Ryan that I'd have GWAS results, but we don't have that yet. So next year, hopefully next week. But I'll talk a little bit about using this internship as a model to find genes and other biomarkers in stress and depression. Using this model also to hopefully help understand and improve medical education and understand how depressive symptoms in general behave and change with time, and then touch on a little bit of work that we're doing with MSIS on mobile monitoring in this population. So I'm a geneticist and a psychiatrist, and in sort of my clinical interest, depression, stress-related phenotypes really stand out in bad ways in a lot of things. This is just one example among the top 10 leading causes of morbidity and mortality in the United States. For almost all of them, as a field, we're making good progress. You're much less likely to die at any age of a heart attack or cancer than you were 20, 30 years ago. The big exceptions are psychiatric disorders, and in this case, suicide. The rates are going up as opposed to everything else is going down. I think one of the big reasons we haven't made much progress is that we really don't understand the underlying biology of our systems and our disease. And one of the sort of hopeful avenues is genetics. As we all know, it's a really exciting time in the last 5-10 years in genetics, and especially with genome-wide association studies. We're finding many more confirmed associations in the tens of thousands of confirmed associations, whereas 10 years ago we had single-digit ones. So there's been a huge amount of progress in complex disorders in general. And in general, there's a pattern with genetic associations where a pretty nice relationship between the sample size of the phenotype being studied and the number of hits that people have found on GWAS. So diabetes and in Crohn's disease and in almost all phenotypes that relationship is there. Depression, which is what we're looking at here, is a pretty glaring exception to the rule. They're up to, I think, 40,000 subjects in the largest meta-analysis of GWAS for depression, and nothing comes out genome-wide significant. I think the largest phenotype that people have looked at without any significant hits. So certainly the genetic architecture of depression is probably playing a role, but that's probably true for a lot of complex diseases. I think two unique characteristics of depression are probably making it harder to be tractable in genetics. So for depression, just like most of the other phenotypes there, people are essentially looking at cases, people who've had an episode of depression at some point in their life versus controls, ones who don't. And for depression, that's really hard to figure out cross-sectionally. This is just a longitudinal study. I'm not involved in it, but they assessed people. They asked people if they had an episode of depression at some point in their life at 4 different time points over 15, 20 years. And the bottom squares are what percentage of people said that they had an episode at that wave, and the top one is the people who said they had an episode of that wave, and added to it the people who reported a history of depression in previous waves. And essentially, you can see by the 4th wave, 3/4 of the people who previously reported an episode of depression had forgotten about it, and now were classified as controls if they were doing a GWAS on this. And this has been replicated in longitudinal studies. So if you have that much error in assigning your phenotype, it's hard to really find a signal. And that's one problem that depression struggles from. Another is just that it's a really heterogeneous disease in terms of causes. So we call someone and classify someone depressed when they, have symptoms after breaking up with a girlfriend or boyfriend in college, and it's the um, same constellation of symptoms and the same diagnosis if after people have heart attacks or strokes when they're um, in their 70s and 80s. So they might look like the same thing, but biologically, they're probably very different phenotypes. And in that way, depression might be something like fever or pain that have many, many different pathways to get there. And if you're just looking at the end phenotype, it's hard to tease out the underlying biology. So that's sort of a long-winded way to get to what we're trying to do with our sample. Uh, using medical interns as a stress model. So we're looking essentially at the first year of medical training outside of med school. This is when residents or interns first have real responsibility. They're working 80 to 100 hours in sort of weird time shifts throughout the course of the year, and often report feeling overwhelmed with making important medical decisions without having enough knowledge or training to make them well. So we look at interns here at Michigan, and also at up to 50 hospitals now around the country. So we enroll them first 3 months before intern year when they're finishing medical school and happy, healthy, and don't really know what they're in for. And then follow them. They start their clinical duties July 1st, and we assess them every 3 months throughout the year. And this is across medical specialties. And we're studying our 9th cohort of individuals right now. We measured depression through this patient health questionnaire, which essentially goes through the 9 symptoms that the diagnostic and statistical manual, the psychiatric diagnosis for depression. So before the year starts, about 3.5% of the interns meet criteria for depression. And during the year, at any given time, about a quarter of them meet criteria for depression. Over the course of the year, 46% of them meet criteria at least once. So it's a pretty substantial stressor and sort of alarmingly high rates of depression. But this lets us bypass some of the problems that previous genetic studies have had. We're focused on current depressive symptoms, which we can measure pretty accurately, much better than we can lifetime history. We're controlling for a whole bunch of personal factors because we're using within subject design and following the same people as they go from the low stress situation to the high stress situation. And everyone is presumably getting depressed in the same way because they're going through the same stress. So, so we've done some candidate gene work. Far and away the most studied gene in psychiatry is this serotonin transporter gene that is sort of the target of the most common antidepressant medication, Prozac, Zoloft. And there's a functional polymorphism, a short-long allele that many people have looked at, and most famously, this Caspi paper found a gene-environment interaction that people with the lower functioning short allele looked like the same as other genotype groups under conditions of no stress. But as I looked at people with more and more stressful life events, the short allele individuals had much higher levels of depression, suicidality. So we replicated and found that interaction in our sample as well, and essentially these are the types of genetic patterns that we're looking for with this sample. With the serotonin transporter, both genotype groups show no difference or the same levels of depression in the low stress period before internship, but people with the lower functioning allele here get higher depressive symptoms during the year. Skip over this. We as sort of an intermediate to doing a full broad scale analysis, we ran this chip of essentially brain genes on the first 1,300 people in our cohort, looking at about 5,000 SNPs, or we pared down to a little less than 4,000 for our analysis. And a couple genes were sort of chip-wide significant. The most significant was this one in SLC6815, which is a neutral amino acid transporter in the brain. Has sort of the same pattern as we saw with the serotonin transporter, and it's actually the one of two genes that have come up in significant in there's been 9 GWASs of depression to date, and this came up significant one of them, but wasn't replicated in any of the other ones. It's in the same gene, but our significant SNP isn't the same SNP and isn't an LD with the SNP that came out in the study. So it's still not clear what the relationship is, but it's interesting that it's in the same gene. Another one that just missed sort of the significance threshold, but also is interesting, is in the fibroblast growth factor gene. That it's a coding variant in the gene that has a pretty dramatic effect on at least, or seems to predict to have a dramatic effect on the protein functioning. And it's in a gene that's been looked at pretty extensively in depression, a lot by Huda Akil and Stan Watson here at Michigan, who've shown that this FGF, the levels of this FGF2 protein are lower in the brains of individuals who committed suicide compared to controls, and injecting FGF2 into rodents early in life seems to protect against anxiety and to some extent depression. So we're following up on that variant and seeing, see if we can understand how it's working more. And we're currently running our first 7,000 subjects on this psych chip so that we can get GWAS results and look more broadly across the genome. So hopefully I'll have those results soon. We're also using this intern model to look at other potential biomarkers beyond just genes, and with other peripheral biomarkers, we can also follow how they change with the year of stress. So one example is cortisol. Cortisol is a stress hormone and has been studied a lot in the saliva and blood, and clearly shows a pretty dramatic response to acute stress. But because saliva in the blood or cortisol in the blood and saliva change pretty rapidly and aren't consistent over time, it's been hard to study in chronic stress. We've been studying hair cortisol through a relatively new technique that's been developed in the last few years. Essentially, your average blood levels of cortisol for the last month are reflected in the centimeter of hair closest to your scalp. So we cut the intern's hair every couple of months and get their hair cortisol levels. We don't see whatever your hair cortisol is at baseline doesn't seem to predict whether you're going to get depressed or not. But for people who stay resilient, who don't get depressed during the year at all, they seem to have an initial increase in hair cortisol and blue up there, whereas the people who get depressed actually stay pretty stable. And at the first few months, we seem to be able to distinguish to some extent people who are going to stay healthy and people who are going to get depressed through this measure. Depression is a pretty strong risk factor for heart disease. People with depression are twice as likely to have a heart attack or stroke at most age ranges. We found that none of these are, our subjects are on average 26 years old. None of them, thankfully, are having heart attacks and strokes, but we can see over the course of the year some decrease in endothelial functioning of your blood vessel lining. And the ones who have the greatest decrease in their endothelial functioning and endothelial progenitor cells are the ones who are getting depressed. So we're maybe seeing the start of the link between depression and cardiovascular disease in these guys. And we're looking at other biomarkers, epigenetic factors, RNA stuff. Happy to talk about that if it's of interest. So this increase in depression is fun for me because it's lets us study the biology of stress and depression, but it's a little bit alarming from a medical education perspective. So we're also doing some work to try to understand why depression is so high and some of the consequences of of um, of the depression in these guys. We're not the only ones to study it. Most other studies looking at it have been cross-sectional, but we did a meta-analysis of all the studies looking at depression during internship and found a similar rate of about 28% across all studies of depression among interns, and since studies have been done since the early 70s, we're actually seeing a small but significant increase in depression over the last 40 years with medical training. We don't really know what's causing that, but it's notable. Yes. Yeah. So I mean, I don't think. Yeah, I don't know what. Yeah, depression is um, uh, a weird diagnosis, but for the first couple of cohorts, we've been following them each year, and now they're 6 years out. So the rate of depression goes down after the year. That during this internship is sort of the peak, but it doesn't go back anywhere near to baseline what they were before. So it seems like and people were depressed during the intern year are much more likely to be depressed a few years out. So yeah. So and as those cohorts get older, we'll get more into the insights into the links between depression and long-term cardiovascular function and other things related to depression. There's a bunch of factors that we have measured at baseline that seem to predict who's going to have high depressive symptoms in the intern year. A lot of them are the same ones previous studies have found for depression in general. And with these factors, we can, and a little bit from the genes and biomarkers, we can predict about 70% of the variance of who's going to get depressed during the year. One of the most or one of the strong ones is gender. Before the year starts, men and women have similar levels of depressive symptoms, and after that stressful year, the levels are about the same again. But during the intern year, the increase in depressive symptoms is about twice as high in women compared to men. There's a lot of factors that seem to be playing a role that we're looking into. One of them seems to be this work-family conflict that correlates pretty strongly with depression. Compared to men, women report concerns and issues with work interfering with family. Men preferentially report problems with family interfering with work, and it's only the former, the work interfering with family, that really relates or correlates with depression. So it's interesting. We don't know what to do with it, but it's there, and also that the links between depression during internship and depression 5 years out, which I mentioned before, and also the link between depression and cutting down to part-time work 5 years out is there in the population, but it's only there in women overall. So sort of this peak of depression in women specifically seems to have consequences that we can detect years out. The sort of governing bodies that run medical education and residency have been trying to change things to make the environment sort of healthier for interns and safer for their patients. And they put in a new rule, 2011, sort of restricting the maximum number of hours people can work to 16 hours, whereas before it was 30 hours. And so we were measuring the same programs, the same cohorts before and after this change. And so people did work a few less hours, 3 less hours a week. They slept like 5 minutes or 8 minutes more each night, so not much of a significant change in sleep. But we didn't see any change in their depression when they cut back the maximum shift to 16 hours. And we saw a pretty dramatic increase, a 20% increase in medical errors. So when they restricted the shift length, the errors actually went up, which was kind of surprising. But it seems like most hospital systems didn't invest or didn't have any money to invest in bringing on new doctors or nurse practitioners or anything to take off some of the workload. So they were expecting the residents were asked to do sort of the same amount of work as they had before, just in less time, which creates a sort of phenomenon called work compression. Might result in more errors, and also there are more handoffs from one doctor to the next. And a lot of information seems to be lost in those handoffs. So it's a really complex system and hard to find solutions. But at least what we're doing so far isn't working too well. The few of these interns actually get any help. About 15% of the ones who meet criteria for depression get treatment, and most of them report that they're not doing that because they don't have time, and there's a lot of concerns. There's a lot of stigma about depression, and a lot of concerns that if they do seek formal help, this will get back to their supervisors, affect their careers in some way. So we did, trying to take those barriers into account, we've done a little bit of working in an intervention. So we did a randomized trial of interns and assigned some to do, before the year started, a web-based cognitive behavioral therapy training, Mood Gym, which is a free website anyone can use, and just takes a few hours to go through and teaches you sort of the principles of cognitive therapy. And the ones who did that, compared to the ones who didn't, the ones who went through the Mood Gym got significantly less depressed during the year and had lower levels of suicidality. On average, 12% of the interns have some suicidal thoughts. So this was a pretty minor intervention, but seemed to have an effect, even though it was administered well before the year started and was relatively non-invasive. So we're working a little more to extend this, and maybe with some of the mobile work, let them have some of these tools that are in this website available to them in real time. One of the in psychiatry and especially at NIMH, the sort of major NIH institute funding most of this research, there's been a push to get away from using diagnoses as our outcome measures. This is sort of how you diagnose depression, and it was constructed mostly for reliability, so that one doctor could communicate with another about a diagnosis, but there isn't any real biological reason these 10 symptoms have to go together or do go together. It doesn't have biological validity. So there's been a movement, and what we've been trying to do is to look at the symptoms separately, and we do see that the different depressive symptoms sort of behave differently. They all go up with this internship stress, but pretty markedly differently. So depression and fatigue go up quite a bit, but sleep, having trouble sleep doesn't change all that much. Similarly, some of the factors that predict, that I went over before, that predict depression during the year have different effects on different symptoms. So having a difficult early life and childhood stress increases your risk of being suicidal and having concentration problems during internship, but doesn't affect your fatigue or mood really. Women have an increase in the mood, fatigue, and appetite symptoms, but men actually have a much greater increase in suicidality during internship. So this sort of speaks to the utility of looking at the depressive symptoms individually. And we're trying to incorporate that into some of our biological work, genetics as maybe more precise phenotypes. And along the same lines, when we look at look at the symptoms longitudinally over the 3-month assessments. I'd originally, or I'd expected that there'd be a sort of mean level of increase in depressive symptoms and a sort of normal variation around that. But when we do growth mixture modeling, the symptom patterns actually fall out in 3 pretty distinct trajectories, where with a group of pretty resilient interns who don't have very high depressive symptoms at baseline and don't really go up at all under the stress, and another group that has sort of medium symptoms throughout, and all the analyses and all the dramatic increase that we've been focused on is driven by one group of about 25% of the cohort who start off low and get really, really high symptoms through the year, the group in blue up there. So again, this is another way we've been looking at the phenotypes to maybe get phenotypes that are more trackable for our genetic analysis. So that's a lot of the work we've done so far. Part of this, like one thing that that followed from that trajectory work is that the longitudinal nature is really important, and that it might be helpful to get more time points and to actually be able to measure these symptoms in real time. So with a small subset with about 40 people, we've used this app to essentially text them every day and have them enter their mood once a day, and it's sort of interesting results. The Daniel Kahneman, who's an economist, has done some work and wrote a really cool book, Thinking Fast and Slow. But one of the things that he got the Nobel Prize for was this discovering this peak end rule. The example that he's used is with colonoscopies. When you ask people how horrible the experience is, it's not a mean of how bad the experience is throughout the whole time. It's the worst point of the colonoscopy and then how uncomfortable it is at the end. So they found that if you actually leave the colonoscope in at the end for an extra half an hour, it's not that makes the end more pleasant, and people rate the whole experience better, because what people are responding when you ask them is the worst point of the procedure and the pain at the end. And the same thing with vacations. When people go on vacation, and you ask them how their vacation was, what they tell you isn't a mean of their overall vacation. It's how good the last day was and the most memorable experience. And we kind of find the same thing with depression with the interns, that when we do those PHQs after 2 weeks, what they're not really giving us, and these are doctors, they're not really giving us a good representation of their mood over the 2 weeks. When we correlate those PHQs with the daily mood ratings, what they're really telling us is their mood on that day, and then their worst mood over the last 2 weeks. So it seems to fit that peak end pattern, and I think speaks to the importance of getting information from these guys in real time. One of the challenges and maybe interesting promises that just getting that information from them seems to be affecting their mood. So the ones that this wasn't randomized, but we just enrolled 38 interns at Michigan in this daily monitoring study, and then we have a whole bunch of other interns at Michigan who aren't part of that study, and the ones who we're monitoring closely, their rates of depression are almost half as much as the ones who aren't. So it seems like maybe just monitoring them is having an effect, which opens up the possibility of relatively easy interventions, but also makes it hard to study. So we're working with these guys at MSIS to maybe develop an app using this research kit framework that Apple's developed to hopefully do the daily mood monitoring, but even maybe some more sophisticated monitoring. There's some evidence that individuals who are prone to depression move around less and are in fewer locations, or are at home much more often than people, and we can potentially use the GPS on people's phones to get that information. There's some evidence that depressed people have less communication, so if we could use the microphones to assess how often they're talking to people, use the sort of activity monitors. A group has developed an app to mostly for commercial purposes, but essentially if you it can use the phone to take a picture of your face as you're watching a video and assess your mood or your reaction, and that also correlates with a group at Duke has been correlating that with risk for autism. It could also work for depression. So we're still working on the details, but I'm pretty excited about this going forward as a way to really monitor people in real time and get objective information rather than sort of our phenotypes right now, which are completely dependent on self-report. And hopefully, if we can get this in a large enough samples, this can feed back to make richer and more meaningful phenotypes for our genetic and biological work. So that was a whirlwind of lots of stuff, but I hope it was interesting, and I'm happy to sort of delve more into any of the parts that you guys found interesting. So I wanted to thank my group and collaborators and funding sources, and be happy to take some questions. Thanks. So, any questions? No?
So the percentages, you said get all the way up to 46% cumulative, but in some of the later studies, it was 14%, 16%. So I'm curious, if you turn it around and ask, what are the characteristics of the interns under similar stresses that don't get these symptoms?
Yeah.
Is there anything interesting there?
Yeah. Yeah, like in that trajectory analysis, about 40% of the sample were in that resilient group that don't budge at all, that seem like they have the same symptoms, and maybe some of them even look a little better under the stress. And so for some of our genetic analyses, we're using that as a target and looking to see if we can find predictors of resilience, being a member of the resilient group rather than the others. So one like the FGF2 analysis, that associates the strength of the finding is much stronger when we look at it the way you're saying, rather than the other way around.
Correlate with resilience?
Yeah, there's personality variables that we measured before. For some reason, people who train outside the U.S. are more resilient. Having children, I think, was so a lot of demographic stuff. And in terms of the biology, we're still in the early stages, but there's certainly a bunch of factors that we know correlate with. We gave them a resilience scale, and sense of humor and religiosity were also factors that correlated with resilience. There's not enough. The doctors scored a standard deviation below the population norm on sense of humor, but.
So I got a question. The difference between males and females is fairly striking, and I think some of the issues that you discussed may make complete sense. But when you're doing your surveys and stuff, how much information do you have in each individual? Can you separate out, say, males and females that are married, that have kids? If you control for that, do those same differences appear? And I'm just curious how far you've had the chance to dig into that.
Yeah, I mean, we definitely have that information, and yeah, it seems for most of the factors, the effects are not different by between males and females. Like so having being neurotic and the training outside the U.S. Having a child especially during the intern year, the dads actually get a little happier, but the increase in depression is dramatic for women. So that having a child seems to be one that interacts with gender for depression. But we haven't found too many others. But there's a couple like
I have my couple cousins who've gone through, you know, the whole system, and they've told me that like the best time they've told to have kids is their first year of residency. And,
Yeah.
And it's me, I'm like, are you crazy?
Yeah.
That seems like that
No,
would
no,
be,
it's not.
but it's just kind of interesting to see that sort of
The
play out.
the other one that came up was the percentage of women in your program. So if you're in a program that is mostly is gender balanced, then the gender difference in depression isn't that high. But if you're in, you know, a neurosurgical program where everyone else is a man, then there's a huge gender difference. So there's lots of those.
It'll be interesting how the genetics turn out too,
Yeah.
because I mean, it sounds like most of this is more environment than it is actual like
Right.
systemic. It'd be interesting.
Yeah.
Other questions?
This is an unusually controlled shared experience, of course. It's a very good choice of design. But have anything comparable to this study been done with graduate students?
No, not to my knowledge. I think it'd be good to do. It's just it's harder to get big cohorts, and at least in my experience in grad school versus med school, it's more of a variable experience in grad school. So if you're, you know, if you have a very generative, good mentor, you could have a pretty nice experience, or it could be if your first paper, if you happen to get a really good publication in the first couple of years, it's much easier. So I think the overall rates of depression are probably pretty high, but the experience, at least in my thinking about it, might be more variable from subject to subject.
Probably right. Notice one of your slides you had maltreatment.
Mm-hmm.
This refers to interns being badly treated by residents and faculty. Is that the idea?
No, sorry, I should be. That was childhood. So being badly.
So
treated
what
by
about
their-
reporting maltreatment as interns
Yeah.
by immediate superiors? There's a lot of literature and a lot of anxiety about this among leadership in medical schools all over the country.
Yeah, and half of the medical students graduating from this institution report that they've been mistreated in some way, and so we haven't gotten to it directly, but certainly the people who report more of that are more depressed. But it's hard to tease out causation. Or I haven't been able to do it in a good way, but they're certainly correlated with depression and also correlated with reporting committing medical errors. So I think it sort of feeds over into affecting patients as well. And I think that as the duty hour rules came in, I think maybe not overt mistreatment, but the interns who are under the new rules were, at least anecdotally, report a lot of resentment from their senior residents who had to go through the old system and now feel like the new people are being coddled and they're having to do extra work that wasn't there before. Oh, sorry. Yeah. Yeah. So I tried to go through it quickly, but yeah, the ACGME is a governing body for residency programs, and there've been a bunch of work like this and high profile actually suicides among interns, so in a series of changes, the most recent in 2011 they put in rules, and that was the rule for 16-hour a 16-hour shift maximum for interns. But they don't, yeah. But as I showed, it didn't, and other work has shown it didn't really have the intended effect. So yeah.
Thanks. Is there any correlation between these highly depressed people or stressed people and subsequent development of serious illnesses?
Yeah. So sorry, I went through it quickly, but a little. There's a strong correlation with cardiovascular disease.
Yeah.
There's a link with autoimmune diseases. So yeah, there's a pretty strong correlation, at least, between depression and a whole range of sort of systemic illnesses.
How about illnesses such as Alzheimer's?
I don't know the percentage. I know depressed people are more likely to
Yeah.
have dementias later in life. I don't know how strong it is, but it's certainly there.
Thanks.
All right, well, thanks very much. It's a great
Thanks.
talk.
Thank you. Yeah.